Hi, welcome to lesson 3.4, skill 3 in action. You may recall the AAMC has identified 4 skills common to all sections of the MCAT. Skills 1 and 2 were all about content and critical thinking. Not terribly surprising for a test like the MCAT. But skill 3 is truly unique. I'll let you see what I mean for yourself. I'm Tyler Farah. Let's begin. To start with, let's see what the test makers themselves have to say about these skill 3 questions. Take a moment and read over this paragraph from the test maker, and in your own words, jot down what they're asking you to do in a typical skill 3 question. So what I'd like you to focus in on is this chunk of text right here. What the AAMC is asking you to do is show that you can, quote, do science. In other words, skill 3 questions are the questions about experimental design, about independent and dependent variables, even about flaws in a given experiment. And so in this lesson, we're going to look at a passage that actually really illustrates how these skills might be tested on test day. We're going to start out, as you will on test day, by looking at the key components of an experimental procedure. We're going to think about the limitations or maybe even flaws that might be present in this experimental procedure. And we're going to be able to also infer conclusions from the experiment that are maybe not necessarily stated outright in the experiment. This passage is going to be a really challenging passage in molecular genetics that also links in the cell cycle and the regulation of certain genes. So without further ado, let's dig into our first learning goal, which is to identify the key components of this experiment. As with every passage on the MCAT, the very first thing that you want to do is preview this passage. You see a figure here about some protein that you probably have not heard of, cohesin, and its ring structure. You see a paragraph that gives some details, some background about the cell cycle. And outside of the fact that I've told you this is going to be an experiment passage, it's important for you to recognize on your own that we're seeing a brand new experiment here. And that information really comes in the third and fourth paragraph, the third paragraph being just a simple listing of steps in this experimental procedure. So since this lesson is all about understanding experimental design, let's break down this passage in terms of its experimental design. First, on the MCAT, you're often going to see experiments being presented with a background paragraph that describes the science. Now, this can be a little intimidating for students because you're going to see, in this case, a lot of stuff that you've never heard of before. This is a new experiment, something the MCAT doesn't expect you to know about. But the key observation is that the MCAT is always testing how you take new information and relate it back to what you do know. So what I'm going to do now is hand this over to you, and I'd like you to read just the information on screen, just the first couple of paragraphs here, but instead of focusing on the brand new stuff that might be unfamiliar or even intimidating to you, I'd like you to focus in on the stuff that you do know, the stuff that you do recognize, and think about how that correlates to your outline. Go ahead and do that right now. So looking at this passage, I see a lot of information about this protein called cohesin, which again is not part of your review notes. It's not something the AAMC expects you to know about. But I see that cohesin correlates to things that you certainly do know about. We see that cohesin binds during S phase. Well, what major event is happening in the cell during S phase? You're thinking about synthesis of brand new DNA. This is where replication occurs. So apparently cohesin binds to sister chromatids during replication, and then we see that in metaphase, the cohesin proteins release, the loops open, and that actually allows the sister chromatids to separate, which is the major event as you're transitioning from metaphase into anaphase. So maybe you've never heard of cohesin before, but certainly you've thought about the idea that sister chromatids are held together from S phase up until metaphase. The new information is that apparently this protein cohesin is implicated in that sister chromatid cohesion. The second paragraph really starts to segue into the experiment itself. And I'm going to turn it back over to you to finish out this experiment and read the details of the procedure. But before I do that, I want to give you a piece of advice. On this experiment or any passage with an experiment, you're going to help yourself out a ton by looking for the independent and dependent variables. Not only are MCAT questions going to test your knowledge of these variables, but in a passage like this, which is really, really complicated, it's going to be super helpful for you to cut through all the details and just look for those big structural pieces, the independent and dependent variables. Take a moment, read through this, and jot down the independent and dependent variables right now. So in this passage, the independent variable, I think actually makes a lot of sense, since our researchers are looking at how cohesin binds from S phase, where we have replication, up through metaphase. What they're doing is they're allowing cells to grow up, and they're taking samples at different times in the cell cycle. This might be a little bit harder to spot than some independent variables because a lot of the times we're used to seeing the researchers causing some sort of change, introducing the independent variable, whereas here the researchers seem to just be letting the independent variable happen. But notice there's an awful lot of setup, a few extra steps that are involved in ensuring that our cells are synced up and moving through the cell cycle at a uniform rate. So the researchers have done a lot to actually introduce and set up this independent variable. As for the dependent variables, I see the keyword down here, results. And remember, the dependent variable is a measurement that generates data from which results are going to be extrapolated. So the word results tells me the dependent variable, what's being measured, has to be close by. And reading on a little bit further, we see that results are generated by isolating and sequencing fragments of DNA, and specifically the thing that we're measuring is where cohesin binds and how many cohesins are going to bind. So to summarize this experiment, stripping away all the details, what we're looking at is a molecule that holds sister chromatids together. That was the point of that first paragraph. And what our researchers are interested in is, as we move through the cell cycle, our independent variable, how many of these molecules bind to the chromosomes and where on the chromosomes do they bind? When you break down the experiment in terms of those structural elements, it seems to make a lot more sense, and it seems to be a lot more straightforward than the details of this experiment initially made it seem. So with that, let's move into the questions and earn some points. Question 1 asks us a really specific detail about this passage. It says, what's the purpose of alpha mating factor? Well, a key insight is you probably haven't studied alpha mating factor, and the AAMC certainly don't expect you to know about alpha mating factor coming into this test. So really what this question is doing is tying something new, alpha mating factor, back to things that are more fundamental, back to things that you do know about, like, for example, experimental design. Instead of looking at this question and thinking, well, this is really about a specific detail, think about how does alpha mating factor relate back to big picture principles like the independent and dependent variables. In fact, take a second right now, go back to the passage, find alpha mating factor, and think about whether alpha mating factor is correlated to the independent variable or the dependent variable. So we find alpha mating factor in the second step of the experiment, which says a protease degrades alpha mating factor, releasing the cells synchronously into the cell cycle. The key word here is this idea of synchronous release into the cell cycle. In other words, it's alpha mating factor and its subsequent degradation by proteases that actually allows the researchers to ensure that the cells move through the cell cycle together. The time in the cell cycle was our independent variable, so alpha mating factor is actually responsible for setting up that independent variable, for setting up the temporal aspect of this experiment. What I'd like you to do now, find the answer choice that seems to match these ideas. Letter B seems to be a perfect match for this. Alpha mating factor release is what sets the 0 time point of the experiment. That's a fancy way of saying alpha mating factor makes sure the cells move through the cell cycle at the same time. As for the wrong answers, alpha mating factor induces cohesin to bind to DNA. Well, the passage tells us that it's actually formaldehyde that causes cross-linkages between cohesin and DNA. For letter C, rapid growth of the yeast culture is caused by alpha mating factor. It's actually kind of the opposite. Alpha mating factor freezes the cells at a certain point in the cell cycle, and only once alpha mating factor is broken down by a protease can the yeast culture continue through the cell cycle. And as for letter D, synchronous release into the cell cycle minimizes intracellular signaling. Intracellular signaling is completely out of scope for this passage. It's not mentioned at all, and that's why letter D is going to be out as well. Similar to the last question, question 2 asks us about a hyperspecific detail from the passage. What's the purpose of sequencing? But again, instead of looking at this as a specific detail from the passage, think about how this correlates to the big picture. Independent and dependent variables. Take a moment, go back to the passage, find sequencing, and think about its role in the experiment. Which of the experimental variables does it influence? So in the passage in paragraph 4, we find that sequencing is implicated in the generation of results. Specifically, sequencing tells us where cohesin binds and in what quantity. So if the dependent variable is the thing that's being measured, in this experiment, it's sequencing that actually generates those measurements. Take a moment and find the answer choice that correlates to this right now. Letter D seems to be a perfect match for this. Sequencing does reveal the chromosomal location of DNA-bound cohesin. A, B, and C are all great tempting trap answers because they are actually all true facts about sequencing that have just been misapplied in this question. Sequencing can be used to reveal the genetic structure of a gene coding for a certain protein. Also, sequencing can be used to map an organism's genome, and scientists actually do this by sequencing individual chunks of the genome and then stitching those chunks together to reconstruct entire chromosomes. This was famously done in the 1990s in the Human Genome Project and actually has been done for many model organisms in the time since. So A, B, and C, all true facts, all things you may have known about sequencing, all misapplied here. And the key insight is recognizing sequencing as the thing that correlates to or actually generates the measurements that are the dependent variable. What we've seen so far is that passages about experiments mean you always know something about the passage because you can always apply principles of experimental design. One of the things you can always spot in a passage is the independent and dependent variables. And what we've seen is that that turns into points. In our second learning goal, we're going to take a look at something else we can always find about any experiment passage. One thing commonly tested by the MCAT is limitations in experimental design. Limitations need not be a bad thing. Sometimes scientists actually impose limitations on their experiment in order to tighten the focus. Sometimes, however, limitations are imposed upon them by limits in technology or limits in the time scale of the experiment. In either case, recognizing the limitations of an experiment is important for scientists, therefore it's important for doctors, and so it's important on the MCAT too. One limitation of experiments is that we try to draw conclusions that are widely applicable to general populations, but we're using data gathered from a small sample. In a well-designed experiment, the researchers will recognize the ways in which their sample differs from the general population, and they'll limit their conclusions based on those differences. So take a second here and think about and record who's the sample in this experiment and the population to whom the results are going to be generalized. The researchers are doing this experiment on yeast, breaking them open, chopping up their DNA, collecting relevant fragments, recording data about those fragments. But why? What's so interesting about yeast? The answer is that the researchers hope to generalize their results to us, to humans. They really want to know, how does this work in people? So they're studying this phenomenon using yeast as a model organism. Question 3 does something kind of cool with this. It directly asks us what limitations there are to using yeast as a model organism. Why might they maybe not be the perfect fit? AKA, what weakens the choice of yeast as model organism? But before we think about that question, I'd like to put this in contrast by thinking about what makes yeast a good model organism in the first place. In other words, in what ways does the sample, yeast, match the population, humans? Take a moment and think this through right now. Jot down some notes about the factors that link yeast cells to human cells, specifically in the context of this experiment. The most key insight about yeast cells versus human cells is that both yeast and humans are eukaryotes. Since this is an experiment about molecular genetics, that's going to have a lot of really cool implications. First off, yeast and humans should actually have very similar chromosomal structures. Second, yeast cells, like human cells, divide mitotically. And since this experiment is all about tracking cohesin binding from S phase through mitosis, that's really important. Also, yeast cells and human cells should have similar molecular genetics machinery. That is, the molecules that are actually involved in the molecular genetics processes should be quite similar. So by studying cohesin in yeast, we can generalize or extrapolate conclusions to cohesin in humans as well. With the similarities in mind, think about a major difference that's going to weaken the choice of yeast as a model organism for studying humans. Go ahead and answer question 3 right now. This question is a little bit tricky. The answer here is letter A, specifically because we are looking at cohesin itself. If the cohesin molecule in humans has different protein subunits than the cohesin molecule in yeast, then that's going to draw into question whether or not the results on yeast can be extrapolated to humans. Well, that's a question that scientists can answer, and in the body of research around this molecule, certainly they have, or they're at least looking at that question. But that is a question that needs to be addressed, and it is a limitation of this experimental procedure that scientists need to be aware of if they're going to draw results from yeast cells to human cells. As for the wrong answers, some of these are actually quite tricky because they are true facts, but they're not actually weakeners. Take a look at letter B. It is true that yeast have linear chromosomes, but then so do humans. So this is actually something yeast and humans have in common and is a strengthener. For letter C, this is also a true fact. Humans have 21 chromosomes, whereas yeast have a different number. But remember, this experiment looked at the relative amount of cohesin that binds throughout the cell cycle, not at the genome-wide differences between yeast and humans. It should be true that if cohesin binds in a certain point of the cell cycle in yeast and releases in a different point, that will be true for humans as well. And as for letter D, again, it's true that yeast are susceptible to alpha mating factor. Human cells are not. However, this is actually something that strengthens the choice of yeast as a model organism, because the fact that yeast cells are easier to work with, more manipulatable than human cells, is actually one of the reasons why we would choose them as a model organism versus human cells themselves. Question 4 also asks about limitations in the experiment, but it does so in a really cool way. It's asking about the factors that would cause someone to doubt the results of the CHIP assay that was described in the passage. So take a second and record your answer to the prompt on screen. What kinds of things cast doubt on an experiment's results? In lesson 3.3, Advanced Experimental Design, you learned about internal and external validity threats. And external validity threats were the subject of the last question just a moment ago. These are things that might cause us not to trust the generalizability of a result. Based on the answer choices to this question, though, it's more about internal validity threats here. These are flaws in the design of the experiment itself. So researchers try to eliminate as many flaws from an experiment as possible, but it's not always possible or actually even necessary to eliminate every flaw. If researchers are aware of the flaws of their experiment and they carefully limit their conclusions with those flaws in mind, then an experiment can still generate useful data. Or the researchers can just try to adjust the experimental setup to fix the flaw. Now, this question specifically is a not question. Or put another way, we're looking at one potential problem that the researchers solve so that it will not cast doubt on the results. So there's actually two possible ways to go about things here, or maybe even a blend of both. You could eliminate anything that is a flaw in this experiment. Or you could look for the one thing that either isn't related to the experiment or is accounted for by the researchers. Either way, it isn't a flaw, and that's the right answer. Go ahead and answer this question now. Now, the passage told us that antibodies are used in this experiment to isolate cohesin. So while it's true that formaldehyde will link many proteins to the DNA, in letter C, we're using antibodies to make sure we select only for cohesin. In other words, letter C represents a potential difficulty with the experiment, but that researchers solved using antibodies. Now, for wrong answers, letters A and B would both mess up the temporality of the experiment. That is, we're hoping to track cohesin binding with time. So we need to synchronize the time in the cell cycle. A and B both mess with that, and so they're out. And then in D, we're actually measuring where cohesin binds. So if we've got some blurriness about where it binds, then that's going to introduce random error into the experiment, which will confound the results. Notice that question 4 also is an application of independent and dependent variables. A and B both had to do with the independent variable. They made it harder for the researcher to effectively apply that time-based independent variable. The letter D has to do with the measurement, right? Measuring where cohesin binds. It's a flaw that should be addressed by the researchers, or it will cast doubt on the results of their experiment. So it's out as well. Only letter C represents a potential flaw that researchers solved, so it's the one that would not cast doubt, and that's what makes it the right answer. So we've analyzed principles of experimental design, and we've also analyzed limitations of a study. The last thing to do is to pull these together and get results. In the third learning goal of this lesson, we're going to consider the experimental design and any limitations introduced by that experimental design, and we're going to draw reasonable conclusions based on these factors. What I'd like you to do before we take a look at a couple of questions that ask us about the conclusion of the experiment, think about the researchers' overall hypothesis and the result of this experiment. It seems that our researchers believe that cohesin is going to bind on S phase and release in metaphase. After all, they hypothesized that cohesin is the thing that holds sister chromatids together. Of note, there is no conclusion given. This is really, really common in MCAT experimental passages. And you might ask yourself, well, then where would we likely see the conclusion of this experiment? The answer is in the MCAT questions that accompany the experiment. Take a look at question 5, for example. Something really interesting happens here. The MCAT gives us a result from this experiment and asks us to draw a conclusion from it. They say, well, our researchers actually did one of these chip assays, and they found that cohesin was bound during G1. This might even be a little bit surprising given the researchers' hypothesis. They thought, well, cohesin is going to be holding chromatids together, so it should bind during replication. Once there's actually 2 chromatids to hold together, that's when cohesin starts to play its role. So what's happened here is there are some additional functions of cohesin outside of the one that this experiment specifically studied, and this new observation is going to support one of these possible additional functions. In other words, given this new data, draw a conclusion. Go ahead and answer this question right now. To answer this question, you might actually ask yourself, well, what happens during G1? And the answer is transcription. Of these various cellular functions, only transcription is actually occurring in G1. So the fact that cohesin binds during G1 may support the hypothesis that cohesin plays a role in regulating transcription. Of the rest of these, of the wrong answer choices, letter A, spindle attached attachment happens during mitosis, not during G1. Replication happens during S phase, not during G1. And genetic recombination happens during meiosis, which is certainly not part of G1. An interesting aside about this question is that according to scientific literature, actually all of these functions are supported for cohesin, but the observation here, cohesin is bound during G1, only supports the thing that's happening during G1, which is letter C. Now our last question is really unique. It's also asking about conclusions, but in a really strange way. Here we have a flawed chip assay protocol, and the question tells us there's a researcher trying to fix this protocol. So being a thorough researcher, she first starts by making sure that she's not the problem. She ensures that she's following the steps of this correctly, and so it's not human error or her technique that's causing the problem. And yet, having made that check step, she still notices that this protocol is not isolating any DNA. So really what this question is asking is, how well do you understand the steps of this procedure? In fact, specifically, remember that sequencing those fragments that have been generated in this chip assay, that sequencing step is what actually generates the results. It's what generates the data. So what we're looking for here is the link between the steps of this experiment and the actual results or the conclusions of this experiment. And again, the twist is one of these steps has gone wrong, and we have to find out which step that might be. Now, I'd like you to try this one on your own. Assess, plan, execute, and answer. But one hint, in the assess step, you're probably going to notice that each of the answer choices corresponds to a step in the procedure. So cross-reference the answers here with the procedure and think about which one of these steps would, or which one of these flaws would possibly cause the problem that this researcher has observed. Now, this passage tells us that fragments are isolated by binding antibodies to cohesin. And so if we're failing to isolate DNA fragments, it makes sense that the antibody might be the problem. Therefore, letter A is the right answer. It might have been just as easy for you or just as helpful for you to rule out wrong answers. So let's see what we can do here. For B, sequencing comes after isolating the fragments. Or put another way, if you don't have anything to sequence because you don't have fragments of DNA, then you can't sequence anything. So B is out. For C, if the yeast don't respond to alpha mating factor, that's a problem. But the problem then lies with synchronizing the cell. If the cells aren't synchronized, but everything else works in the experiment, then you'll just have some yeast in S phase and some in metaphase, and so you'd still see cohesin binding, and you'd still get fragments that could be measured. You just wouldn't have useful results. And then for letter D, that's actually the exact purpose of formaldehyde in this experiment. It's meant to permanently bind cohesin to DNA. In fact, cohesin does get removed from the DNA later on, but the passage tells us that's done by chewing cohesin up with proteases. So cohesin never actually unbinds from DNA, it just gets destroyed. Therefore, letter D is out because, again, that's the whole purpose of formaldehyde in this experiment. Overall, this lesson really reviewed all the learning goals from Chapter 3 as a whole. If you want to look at a brand new experiment, the first thing that you should think about is how that experiment relates to the scientific method and what the independent and dependent variables are in an experiment. You're going to see a dozen, maybe 24 experiments on test day that you've never seen before. But I like to tell my students, you know something about any experiment that's being tested because you know the scientific method, and if you can practice this and treat it like a skill to study, you're going to have that advantage on test day. Also, the MCAT has specifically called out particular critiques or flaws of a specific scientific experiment. We looked in this lesson at matching the sample to the population. We also looked at some aspects that might cast doubt on the association between independent and dependent variables. And lastly, the MCAT expects you to recognize how valid conclusions can be drawn from research, or even how New observations might generate new conclusions or change conclusions that have been drawn. The biggest takeaway from this lesson is that whenever you have a passage about an experiment, you always know something. You can always identify the dependent and independent variables. You can always find limitations or potential flaws in that study, and you can always look for the hypothesis and the conclusion. Don't be intimidated by complex or challenging terminology. Instead, look for these principles of experimental design and trust that the AAMC will reward you for your effort.